Okay, welcome everybody to the lecture today. I'm Peter Schwartz. I'm the director. I see of the IU Center for Bioethics, and it's my pleasure to introduce our speaker today, Dr. Suzanne Haga. I think I'm getting that right. It is Haga. Good. And so, as many of you know, we at the Center for Bioethics often host seminars on topics of interest to the university and to the Center for Bioethics, areas of research expertise and work being done here in the area of ethics and communication. And we're thrilled, actually, that Professor Haga agreed to come and speak to us today on a topic that's very close to the hearts of many of us here working in genomics and precision health and pharmacogenomics with a leading group in our department here. And so Dr. Haga has done excellent work in this area on communication and ethics of pharmacogenomics. I would really call it the best work out there. And we're thrilled to have her here to speak about these topics. She is an associate professor of medicine at Duke University and an executive team member of the Duke Center for Applied Genomics and Precision Medicine. And so we're thrilled to have her, and Dr. Haga.
Good afternoon, everybody. Okay. Great. Well, thank you so much for the invitation to be here. I've never been to IU, so it's always nice to see something new and get off of my own campus and see what the rest of the world is doing, especially in the area of pharmacogenetics. So I am a human geneticist by training, a molecular geneticist, and I quickly realized I didn't want to be in the lab, and it was at a time when the Human Genome Project was going on. There was so much interest and excitement and anticipation of what the future would bring once this project was done. So quickly migrated out. Did I just go off?
No, you're good.
I just quickly migrated out of the lab. I did internships and.
Another sample. I gave my opinion and brought her in and said, make sure she agrees. Where's it coming from?
Okay. So I began to get some bioethics training and policy training on the job through the law school. I went to the University of Maryland, so I was interned in Annapolis and Washington, D.C. And this was at a time when everyone was trying to pick up and understand what are the policies, what are the regulations to lay the groundwork for introducing genetics and genomics into the clinic in particular, as well as in other areas of industry and society. So I do a lot of undergraduate teaching. These are truly undergraduate. I teach freshmen, so 17, 18-year-olds new to the campus. I teach a course in bioethics and genetics and genomics research, and so that's a real eye-opener for them. I also teach an upper-level bio course, so I got an appointment in biology as well, and this is more hardcore technology-based. So we go through everything from DNA sequencing, RNA sequencing, microbiome, a lot of the nuts and bolts that they have read about, but they have no idea about the technologies and what goes on in those laboratories that are actually handling the samples and processing them, and they just get a file at the end. There's a lot of work that went in behind that. So we talk about that as well as some of the applications. So this is not a research-based talk. I do have research going on in pharmacogenetics, specifically with community pharmacies and understanding new delivery models and implementation. So I think about that a lot. So this kind of came out organically of discussions with within my class about where does PGX fit in? Is it more part of the human genetics domain where I grew up and was trained in, and we have genetic counselors and clinical geneticists and medical geneticists, or has this permeated the spectrum and is no longer a genetics subspecialty? It is truly a part of medicine, as lots of laboratory medicine is, and it's becoming more and more standard. So this is kind of where this talk kind of originated and grew out of. So I apologize if it's more of a storytelling, kind of a vision experiment instead of a hardcore research, but maybe this will give you a little bit different flavor from the types of lectures that you typically go to. So that is my background on how I came about this lecture or this talk. So I know from my own experience in running forums, people have to leave, so I'm going to give you the conclusions first. I would never do this in my class. I lose all my students. But just to kind of lay the groundwork of where I'm going and see if I can convince you this is sort of what I see as a vision. It certainly is my opinion. I'm open for discussions about where pharmacogenetics fits into the big picture of precision medicine or just the general practice of medicine. So many times people equate precision medicine with DNA-based medicine. It's molecular. Before you need it, rather than to delay treatment or to have to adjust treatment once you get that test result in, since things are not as quick as we'd like them in the genetics lab yet. And as I said, PGX is not an independent test. It needs to be used in parallel with lots of other tests that can give a more broader perspective of the patient's phenotype, these functional-based tests of looking at maybe gene expression or microbiome, and so I'll go into that. So in order to kind of deliver this vision, I would argue, and I've always argued for more patient engagement. However, this is a complex area. Genetics and genomics, which we rarely use in talking to patients, is tough. And the question is, how much do they need to know to actually understand and to participate in these types of assessments or types of care? How do we communicate complex information? We have lots more tools than we had 20, 30 years ago. It still doesn't simplify the complexity of this information, but maybe they don't need to know they're a star 1 or star 2, whatever the case may be. That's not going to impact on health outcomes. You know, how do we figure out what information will resonate most with patients and providers that will result in the largest change in health outcomes? So, as I said, this kind of originated in a lot of discussions when I go through a lot of technologies, and I go through lots of examples that are in the clinic now, as well as those that are up and coming, that are mostly in the literature and research studies. So I would start with some of the major public health issues that are facing us. One is obesity and overweight. The second is cardiovascular disease, and the third are medications. And so I will briefly describe those, but more focus on the genetics, in particular the microbiome, specifically the gut microbiome, and a little bit more on environment. But really the first two and how they impact all three of these and where PGX fits into this puzzle. So this is not new, especially as we're inundated with lots of information from the public health sector of late. But one of the major issues is obesity and weight gain. So overweight and obesity is to the 21st century what infectious disease was to the 20th century, and we might have to change that. But so the prevalence has increased. These statistics are everywhere, and it's not limited to adults. And that it takes a huge toll. Personally, individually, economically, societally, there's clear ramifications any which way you look at the data. And there's no clear single solution to reducing this problem. It continues to challenge society, state governments, Medicaid, Medicare, whichever part of the sector you're in, there are issues related to overweight and obesity. So as I said, it continues to climb in numbers. It's concentrated in certain parts of the country. So these are lots of CDC statistics that are available from 2011 to 2018. You could see a tremendous change in the proportion of individuals that are in the BMI of over 25 or over 30 for the overweight and obesity categories, particularly concentrated a lot in the Southeast. It affects all races, but there are some differences in prevalence between race, by states, by education and income level. So the data's been cut many ways, and there are trends that are obvious, but you can see the maps here, self-explanatory. So it goes right into when you look at the morbidity and mortality lists that every country generates. And for the United States and many nations, diseases of the heart continue to be in the first three, if not the first. I keep going in and out. The first cause of death in most countries. And so these are some of the latest statistics. This compares 1980 to 2017. In general, these lists are pretty much the same. The order has shifted around, but diseases of the heart remains number one. And you can cut it by race. It's still in the first or second categories of causes of death in this time frame. So African American, Hispanic or Latino, it's number two. It's the Asian and Pacific Islanders, it's number two as well. So this remains a huge issue, and it will likely be exacerbated as we continue to face individuals that are coming of age that will face increased risks for diseases of the heart due to their weight. So bringing together the first two, obviously, lots of risks are increased due to overweight or obesity. The one that I will focus most on is CVD or cardiovascular disease, but it's not unique that these individuals with obesity face a lot of issues, and they will likely need a lot of medications. And medications in individuals with overweight or obesity issues may behave differently. You're going to have different pharmacokinetics, and you're going to have different pharmacodynamics. And so that's another issue to factor into how to improve treatment outcomes when dealing with individuals with these issues of heart disease and overweight and obesity. So the third issue I wanted to raise is medication use. So most of us are taking a drug. So about 70% of the population, adult population, is taking at least one prescription drug. 1 in 5, so a quarter, 22%, almost are using at least 5 prescription drugs. So this issue of polypharmacy, and it's always amazing to me when I work with the cardiology clinics, care clinics, that's a time-consuming part of office visits. This is drug management, and these individuals, 6, 8, 10, and more. And it's daunting to the prescribers as to how to balance it, especially if we don't have team-based care and have a pharmacist on hand that can provide a deeper dive into their medication history and really assess adherence and other issues. So prescription drug use has been increasing with age. I have another slide that shows it's actually on the downward trend, which is a good thing. Chronic illness, as often affects patients with obesity, as I said, requires long-term use of medications. So if you look at the top drugs that are being prescribed, this is of last summer, and divided by 2 age groups, 40 to 59 and 60 to 79. A lot of these are related to diseases of the heart and cardiovascular disease. So it's not surprising that this is what we see, given the other trends of, that this is a major health problem facing our society, and it will continue to increase. But there are lots of drugs being prescribed, and patients are taking typically more than one if they are dealing with these issues. So as I said, the trend had been increasing if you look at the first part of the century from 99 to 2008. And these are significant increases from one or more drugs from 43 to 48%. And then the polypharmacy up to almost 11%. However, good news, I think it is slightly plateauing, or it's beginning to decline. So in the next set of data that's available from the CDC from 07 to 16, there is a drop from that 48.3% that was in the other slide to 45. That is not significant. However, there are some significant declines in children and certain, I think it's the 60 and over, which is declining. I'm sorry, it's not significant. So we are seeing a drop off in children and meds. The others, it's trending downward, but it's not yet a significant change. But where do people get drugs? Most often they're office visit. Your first stop is primary care, and those are the docs that are prescribing most of the drugs. So about 3/4 of the visits, you're ending up with a prescription. What are they prescribing? Those are the top 3 drug classes that they're prescribing. The other is the ER, the ED, or you're going to walk out with some type you've already treated, and you're probably going to walk out with a prescription. So 80% of the visits involve some type of a drug therapy. Again, it's analgesics is a little bit different given the differences in urgent care. But these are where drugs are being prescribed the most. As I mentioned, polypharmacy remains a problem, and it's continuing to climb. It's higher in women than men. So the curves at the top are for at least for one drug, and then it's 3 or more. So we have a society where lots of people are taking lots of drugs. And so we introduced the drug-drug interaction, and then I'll introduce the drug-drug gene or drug-gene interaction as well. But there are other factors at play that I'll bring in that may dilute the effect of pharmacogenetic-based testing or DNA-based testing in trying to understand drug response or predict drug response, specifically with respect to adverse drug events. So that is, you could say, problem 2A. Excuse me, that would be 3A. That's a significant issue, that there are lots of hospital admissions due to adverse drug events. Who? Mostly older individuals. So if you're the older adults, so that's defined as 60 plus, 7 times more likely to be hospitalized due to an adverse event. And nearly half of those were in the upper age group of 80 or more. So most of these hospitalizations due to adverse events are unintentional overdoses. So confusion, taking the wrong drugs, so perhaps more often than you had recalled, and things of that nature. The top 4 classes that cause these adverse events that lead to hospitalization, probably no surprise, the warfarin is your first one, and then insulin or antiplatelet agents and hypoglycemic agents. So most of them are related to diseases of the heart or cardiovascular disease. So these will continue to be a major issue any which way you look at these conditions and these drugs. Excuse me, my typo again. So I guess the second part of the medication issue, the adverse event being one of them, is the adherence. That it's well documented that for certain conditions, especially cardiovascular disease, that people don't take their drugs as prescribed. So they don't follow the regimen. Some of them don't even fill the prescription once they leave the doctor's office. And that could be for various reasons, and there's lots of literature on that. They don't take their medication after they fill the prescription. They stop taking the medication. You know, they don't feel sick. You may not feel sick from having high cholesterol, even though plenty of evidence that shows you need to get the cholesterol under control. Or maybe they cut the pills in half. You have an economic issue at hand. That patients are trying to stretch out that bottle of pills, whatever the case may be, but they're not taking it as prescribed, and therefore it may probably not work as prescribed or intended. So there are safety issues related to poor medication adherence as well as poor patient outcomes. It's been quantified in several papers. This is just one from 2011, so a bit outdated, that it's costing a great deal, and it affects a great deal many patients that they are not adhering to the prescriptions when they are being prescribed. They said there's lots of reasons. It could be an unintentional, you just forgot to bring your pills, you forgot to take them to work because you have to take them at lunch or whatever the case may be. Some of it's intentional. You don't feel like you need them. You're fearful of the side effects. Money is expensive, so you're either going to cut the pills in half, or you're going to take it every other day, or only take it when you don't feel well. So there's lots of reasons why we don't adhere as to the prescription as written out for us. But it all results in potentially poor health outcomes and increased costs in the long run. So kind of bringing it all together, so we have obesity plus CVD plus this increased use of medication, potentially increased risks for adverse events, and potential poorer outcomes when their medication's not being adhered to. So obesity-related conditions, so it's sometimes abbreviated as ORCs in the literature, require more medications. So that is heart disease, pain issues, psych disorders, and asthma are the top ranking conditions. As I mentioned, there's altered pharmacokinetics and pharmacodynamics in patients with obesity, and that's something that certainly has to be taken into consideration as it's being prescribed. And it's just another part of the complexity in reaching an appropriate dose that's going to be effective for that patient. There was a study not too long ago, I apologize for leaving the reference up, off in Geisinger, which is a healthcare system in Pennsylvania. So about 57,000 adults looking at electronic medical records, chart reviews, mean age of 47, BMI 29. I'm looking at obesity-related conditions, and they span the gamut, most common being dyslipidemia. It's dyslipidemia, hypertensive diseases, osteoarthritis that were most expensive to treat, accounting for over 18 million as they calculated. And those with hypertensive diseases and osteoarthritis were most in individuals with obesity than those without. So there is a difference in the care spectrum, the dosing, the medication usage, and in full the cost to treat patients with obesity. If you look at the top prescribed medications, there are several groups that put out lists annually of medications that are most prescribed. There are several that are related to diseases of the heart. There are several, now we're getting into pharmacogenetics, that have information that certain genes affect the pharmacokinetics in particular for these diseases. Pharmacodynamics would be more on the cancer side, not all of them exclusively, but as you can see, there's only one that has, it's maybe a little outdated, a guideline. This is from the CPIC group, which has a series of guidelines that they publish regarding the evidence to support adjusting dosage or drug selection based on one or more genes. And so that's abbreviated as CPIC, and that was for simvastatin number 8. So we anticipate that the list will grow as we understand more about some of the pathways and genetic mechanisms behind, but it's still lagging with respect to professional guidelines about how to use these tools or these types of test results in the prescribing of these medications. So where does genetics and genomics fits in? So this is where I live most of the time in trying to look at these new technologies and applications as they come online or are being developed, and as they are deemed ready for clinical implementation studies and eventually, hopefully, clinical implementation to improve care with respect to, in my case, medications where I focus most, but it actually is everywhere along the disease spectrum or the healthcare spectrum that we can find a home where genetics and genomics is involved. And actually, it's a lot more complicated than just the DNA-based or the molecular analysis that you have a certain SNP or a single nucleotide polymorphism in one gene that we know is associated with one drug. It's kind of old school. It's a lot more complex, as you can see, and I'm trying to figure out how to depict this or illustrate this. It's probably unsuccessful, but there's lots of factors that ultimately affect health outcomes with respect to medication use. So if you back it up to it's just your diet, and actually I have another slide that starts before your diet. There's lots of factors that involve or impact your risk for obesity and weight gain. How likely you are to struggle with maintaining a healthy weight. So we have genetics as in the form of nutrigenomics, nutrigenetics. I'll go into that a little bit. It's actually more complicated than just fat metabolism or carb metabolism. There's some different subgroups of nutrigenomics that could affect food preference, food choice, and then weight gain. The other part of the genetics of the diet to obesity relationship is the microbiome. And so this is relatively new in our genetics and genomics world, though it certainly has picked up tremendous speed as we recognize that the microbiome, and there are multiple microbiomes on our system. I'm particularly interested in the gut microbiome as it relates to obesity and medications, is looking to play a significant factor in how we metabolize medications and our risk to diseases. I will keep coming back to medications since that's the heart of the talk. But It starts way in the beginning, and it starts at birth, essentially. So moving between the connection of obesity and certain disease risks, there's a great deal in genetics, in particular on disease susceptibility. Who is susceptible to developing coronary artery disease, or who is more susceptible to developing a certain type of cancer? So there's a great deal of literature, and that remains a huge issue in trying to sort out the impact of a gene, or actually some multiple genes, and trying to understand risk assessments, so these polygenic risk scores. There's a lot of interest in trying to figure out what's the combination of certain genetic variants that will increase or predispose someone to a higher risk of a certain disease. And again, the literature is full of animal models, as well as beginning to see more human-based studies of the connection between involving the microbiome, as it may increase one's risk of certain diseases, in this case cardiovascular disease. And once a disease is diagnosed, the treatment or the intervention, so again, medications, it's the microbiome coming back, as well as pharmacogenetics and pharmacogenomics now, that understanding that certain variants in these genes, particularly liver enzyme genes, but there's also transporters and other genes that are involved in these metabolic pathways with respect to medications, particularly in the liver, are important. To what degree are they important? And I think that's still being sorted out. We've gotten a lot of the low-hanging fruits, more of the one gene, one drug association. And it's trying to work out adding in everything else, and how do we come up with a better predictor of whether a given medication is going to work for someone for a given dose and a given BMI or a given disease, and pulling all those factors together. So as I said, genetics is, you don't have to look very far. It is everywhere, but it's in different forms. So whether it's host or microbiome. So with pharmacogenetics, one could argue that it actually overlaps with a field called nutrigenetics or nutrigenomics. So a lot of the same genes are involved in metabolism of xenobiotics or transport. They're both expressed in the intestine as well as in the liver. And so they have multiple functions. It's not surprisingly a lot of pleiotropy of what these genes do, and we still don't know what many of them are in with respect to fatty acid metabolism or carbohydrate. Then you put as a backdrop the microbiome. So it's not just the composition of our gut microbiome, it's also what they're producing. So introducing metabolome, or the metabolites that are present in our system that may impact how we are processing medications. It may impact gene expression. So the complexity is daunting in trying to bring together these different types of data and trying to assess one's likelihood to respond in a positive manner to a given drug. So a lot of this harkens back in human genetics is biochemical genetics. And that's where I started with interest in genetics in particular, and working in the biochem laboratory. And this, those of us that were trained in human genetics, I'm one of the fathers of genetics, noted way back in the 1930s that there are differences in how people respond. There are different phenotypes in how people respond to xenobiotics, including drugs. So this was not unbeknownst back then. It just wasn't clear as to what affected these differences or accounted for these differences between patients. That may be very slight differences. You just need a little bit higher dose, or it's a mild nausea, to something that's pretty significant, if not fatal. So if you look at the field of Mendelian genetics, there is some pharmacogenetics in it when you go back mid-century. So some of the early examples of these are more the single gene, single drug combinations, and the low-hanging fruit that we've discovered. So with G6PD, it's an X-linked gene. This was discovered in the 1950s, and so there were differences in how individuals process primaquine due to these red blood cell defects because of this mutation in this one gene. Pseudocholinesterase, again, in the 1950s, is an autosomal recessive disease. So you can actually track that through families, but severe outcomes for this drug. So human genetics is inherently family-based genetics, and so go back and actually see pedigrees for drug response. And see how the disease was transmitted, and be able to ascertain if it was recessive or a dominant effect. So another well-known disease or drug susceptibility or adverse event is malignant hyperthermia. Again, from the 60s and 70s, it was identified. that individuals exposed to certain anesthetics may have some severe outcomes. This is a gene, the RYR1 gene that is still current, I guess, in our literature today, is that it's on a list of genes that we actually return the results to for patients that undergo whole genome or whole exome sequencing. And it's, I think, the only pharmacogenetic gene at the time, or at this time, that's on that list. This is an autosomal dominant effect. And so even back then, you have the likelihood of developing this phenotype, but it's actually a susceptibility phenotype. We don't describe pharmacogenetics as a susceptibility necessarily. You don't usually see that in the literature, but this is a drug adverse response susceptibility risk. And so it's the prevalence in very simplistic form. It's the prevalence of the allele times the likelihood, prevalence or probability of being exposed to the drug and having the probability of that adverse event. And so we all carry variants in lots of our liver enzymes in particular, since they're so hypervariable, that we may never express, we may never manifest, simply because we're not exposed to those drugs. However, since a lot of those enzymes are important to many of the medication groups that are commonly prescribed today, that risk goes up, that we will develop an adverse event. It's still a risk, that there are lots of factors at play in figuring out what's one's risk. And so we don't have data. We don't have a lab result or report that's going to say you're at 35% risk because you have the presence of a certain variant and a certain gene that once exposed, you may have that condition or that adverse event. So it's gotten, it's certainly what we would call a complex phenotype or a complex disease, and that many factors are at play. Let me just switch briefly to nutrigenetics. This has been around also for a while. My memory would be, say, the 1980s, when there was understanding of lots of things that affect our food choices and our food preferences and how our food urges. Lots of the hormones related to hunger, that we have lots of animal evidence, less so in humans, that there are many, many genes that affect many, many parts of diet, consumption, processing, metabolism, absorption, and the like. And so these are just some of them, and they do overlap with the genes that are involved in medication metabolism. As I said, these genes are expressed mostly in the same places, liver and intestine. And so there is overlap, and some CYP genes do process fatty acids or carbs, or actually more fatty acids. And so you can look at lots of tables, lots of charts, lots of review papers that are putting together the gene families that are involved in oxidative processes or transports or transferases and so forth. And these do overlap between the field of pharmacogenetics and nutrigenetics, kind of tying those two groups together, that they are the same gene families that are involved. And so you can go into any of the databases, and how much familiar you are, we do a lot of this work in my classes, and we go into do a deep dive for a given gene. So this is CYP2D6, and you can see it has lots of functions. It's involved in lots of metabolic pathways with respect to medications, but it has other work that it's doing. It's not just waiting around for you to take a medication before it's activated, that it's involved in fatty acid metabolism and other processing of other substrates or xenobiotics. And so all this data is readily available, and you can pull up any single gene and begin to see the connections between certain genes and certain metabolic processes. And so just looking at, this is another database, it's called the Reactome. You can see lots of pathways involving cytochrome P450, which is one of the major families of enzymes that are expressed in the liver, that we know in the pharmacogenetics literature is very important to how we metabolize medications, but is also very important for other pathways, other xenobiotics, and in particular food, that these may have an effect. So, as I said, it only gets more and more complicated, not less, unfortunately. So when you add in the gut microbiome and begin to look at some of the literature of how important this organ system is, I guess you could say, to the processing of medications, it's daunting to sort out. And so these have given rise to subfields. So we have pharmacogenetics first, which had an umbrella name of ecogenetics. So now we have, with the rise of the gut microbiome and the field of metabolomics or the metabolome. We have pharmacometabolomics and pharmacomicrobiomics. So imagine discussing these with your patients. As they get more and more complex and we're seeing these interactions between medications and how they may result in a different outcome given differences in our gut microbiome, which we know to be extremely heterogeneous. So and the connection between the gut or the intestine and the liver is critical for medications as well as foods. And so there's that overlap between nutrigenetics and pharmacogenetics and drug response. This is different. So there are lots of differences that have been identified with individuals with obesity and overweight, and that lots of factors can affect the composition of the gut microbiome, such as surgery or weight loss surgery, bariatric surgery, individuals that undergo that have a different microbiome pre and post, and there's evidence that shows the differences upon surgery. They have a different gut microbiome composition, how that may or may impact medication use and other things, it remains to be seen, but there's lots of ways to temporarily or somewhat permanently change or alter the gut microbiome that may have long-term implications for other issues that these individuals may be facing. So it's a diverse, there's 5 phyla, bacterial phyla specifically that are expressed in the gut microbiome. Individuals with obesity actually have less diversity. So that's before surgery or any other medication, you just see that phenomenon, a difference between individuals with obesity and without obesity. And there's lots of literature to support these changes in the gut. So again, more complexities, more loops going everywhere, or arrows going in every direction. With the gut microbiome, this is something that starts from birth, and understanding different delivery options, whether it's a C-section or vaginal birth, you likely will have a different microbiome at the outset. It may even off. Usually by age 3, you resemble more of your adult microbiome, that it reaches a stability. Breastfeeding, formula, all those things go into shaping your gut microbiome, and whether or not that may pose the child to be at risk for childhood obesity. Differences, layer on differences in genetic variation that may affect diet, metabolism, food choices, and so forth, may affect risk of obesity. And then, when a disease does manifest, or the implications for medication use, the importance of the gut microbiome, and even surgery can alter some of these, the gut microbiome, which may then have effects on the medication use. So as I said, lots of things can, unlike the DNA and the host, lots of things can impact the gut microbiome. Some of it's temporary, some of it is more permanent or over a longer period of time. So we can do lots of things to modify it, whether intentionally or unintentionally. Small differences may result in large clinical impact. That remains to be seen. As I said, there are 5 major phyla. There's bacterial presented in the gut microbiome. There's an Irish team that published a few nice reviews with beautiful figures, so I'm going to just borrow their figures, that lots of things can impact the diversity and composition of our microbiome. So obviously, what we ingest, what we are taking medication-wise, our age, our gender, also can affect the microbiome composition as well as the metabolome. And actually, that may be the output that is more critical to understanding drug response and how this may affect gene expression in our own, in our cells, not the microbial cells, and how that may then affect drug metabolism and our risk for adverse events. But again, this is such a dynamic field that every a new publication about something else that impacts the metabolome, and doing series of experiments, small population, and understanding, just taking snapshots of the microbiome and charting how it changes over time, given exposure with certain interventions. A Mediterranean diet versus a Western diet, there are differences in the microbiome. Male, female, there are some differences. How relevant they are to clinical outcomes or medication response still remains to be seen, but there are clearly differences. So kind of bringing it back that, where does understanding that you're a CYP2D6 poor metabolizer fits in? Does that even fit in? Has that bubble actually shrunk given everything else that's going on that may dilute the effect of a pharmacogenetic variant? I think it's part of the predictive response, and we just don't know. We have lots of puzzle pieces. We just don't know how they all fit together and how to calculate more accurately one's response, particularly given an individual with obesity and cardiovascular disease, a woman, a certain age, a certain culture, a certain diet. How do you actually interpret that pharmacogenetic response or that CYP2D6 star 4 allele, whatever the case may be, in that context or in that backdrop of that patient? So this is probably way redundant given the times that we are living in now, but kind of a public health 101 lesson that there are usually 4 stages in general to understanding a public health event or an issue and how it's approached, and that surveillance is usually the first step in trying to understand what is the problem. Risk factor identification, what's the source of the problem, what is the cause of the problem, what's the vector, what are the interventions, what actually works, is it a treatment, is it prevention, and understanding the evidence for that, and then actually implementing it. How do we implement it, especially on a large scale? So how do you do it? So we all get a lesson again in how do we wash our hands, and that's believed to be one of the most effective interventions is hand washing, and it's something as simple as that we are now all singing happy birthday as we wash our hands twice, and teaching our kids, you need to be washing your hands a bit longer because that's the major intervention. You can apply the same approach to any health issue and sort of apply it to genetics and genomics, and that we are kind of stuck in this intervention evaluation of what works and what doesn't. We call it clinical utility. Insurance companies may call it something different, but that's sort of where we're kind of living in the moment, as well as risk factor identification. I know your genes are important. I know CYP2D6 in particular is important, but I also know it's not the whole picture. You could still see a wide range of clinical response or heterogeneity in an individual, that we aren't certain of the pathways and other factors that may impact certain less or more complex non-Mendelian events, that it's not just one gene, one drug. And so the public health roles is you spend most of their time in developing the policies of how do you actually implement the intervention, and assurance. Are people getting access to what they need to address this issue? So working it from the problem to the solution. So having grown up in this Human Genome Project era, and then it's precision medicine, well, I started with genomic medicine and personalized medicine, and then precision medicine, of using more and more data to inform medical decisions and medical management, including the use of medications. Pharmacogenetics was seen as the low-hanging fruit. This is the easy stuff, quote unquote. And for some cases it has been, that it's a very clear relationship. Nothing is 100%, but there are some examples that we have, the community has put forth, that if you have this genetic variant in this gene, recommend lowering dose or switching drugs. This is not for you. And there's enough examples in that that we've all become excited and encouraged that this is an area that we want to move forward in, and that we should move it from a reactive response to testing at the time of medication need to a more preemptive implementation model, where you're getting it right off the bat, potentially at birth. Lots of children, as I said, are getting more and more drugs. This is something that could be useful. However, we also now acknowledge that there's lots of other factors that are at play that may reduce the impact of a genetic variant, or may have different responses in tandem with that genetic variant. How do we factor all that in? So this is either a dream or a nightmare of the data scientists and big data, and how do we pull all this together to actually make more sense of what we can actually accurately measure now, and have some understanding of how it may work, but that's without considering so many of the other factors that go into play. And maybe we're getting way ahead of ourselves, that before that low-hanging fruit was there, it was a flower. And so my analogy of backing up to starting with the risk factor, in this case it was overweight or obesity, understanding that better, and then getting to the actual intervention at the end of the spectrum and trying to identify better drugs or more appropriate dosing based on the patient's genetic makeup with respect to certain genes. So as I said, PGX lies in this area, sort of at the bottom of the spectrum, and there's lots of different interventions. These are three major ones in particular as it relates to obesity and heart disease, surgery, therapy, or behavioral modifications, and there's lots of factors that are going to affect the success of any one of these, in particular in the world of genetics or pharmacogenetics, and now in the microbiome sphere. And so, historically, you see patients that do not respond well to certain medications. You see that it runs in families. We can identify a gene. Again, that's a low-hanging fruit of pharmacogenetics, and so that has spurred lots of discoveries with respect to pharmacogenetics and understanding the one gene and one drug association. Add in everything else, and it makes it so much more complicated, but perhaps will inform decisions better if we had a little bigger picture assessment of the patient, starting before they are in need of medication, before the diseases actually manifest, and trying to address this. And genetics and genomics is everywhere. So how do we integrate pharmacogenetics along with some other types of genetics and genomics and other clinical assessments into understanding and getting a better drug response profile at the end? Loops everywhere. Everything affects everything. And we tried to think of, draw all the lines, and it just ends up with a big mess. But there are, everything is interconnected for the most part. You can draw a line between any of these boxes and find a relationship somewhere with some type of level of evidence. So, we can divide those tiers, if you will, of being in a risk factor state or at risk for a disease, the risk factor being weight and obesity, the disease being cardiovascular disease, the interventions, multiple interventions. But the point of the slide is that there's lots of measures that we do take currently and that we may take in the future. Some of it is coming on board. So on the far left is with standard clinical measures. We look at a whole host of factors that may impact one's risk for a disease, and then which factors are relevant to understanding which is the best course of management. Is it surgery? What type of surgery? Is it a medication? Which dose? Is it a combination of the two, and so forth? And so we are continually assessing these clinical measures, and that's pretty much standard of care. And so adding on to the next layer where we are kind of living right now is understanding the risks based on or due to genetic variants. So this is our DNA looking at risks for obesity, any of those different pathways, whether it's hormonal or it's metabolism or transport, fat storage, you name it. There's lots of factors that go into play there. Looking at risks for certain diseases. So there are genes that have been associated with different types of diseases of the heart, and then once diagnosed, what are the one's risk for developing adverse events for a given drug class or a specific drug. So as I said, most of this is DNA based. We're looking at sequence variation or copy number variation in certain genes that have been associated with any of these conditions. Some would argue that we need to move beyond it because DNA is not a reflection of what is actually going on in the system because it doesn't take into account all those other things, such as the microbiome, your environment, do you smoke, what's your diet like, that perhaps a gene expression profile would be more accurate. Now that's extremely dynamic. You could take 4 samples during the day and get 4 different data sets, depending on what your environment is and what's going on. So that's very different than what we would get from the one-time DNA-based test of identifying variants that you presumably will have for life. Changes can occur. The same with the microbiome. This is a dynamic data set that you could take it every day, and it may look a little bit different depending on what's going on, the time of day you assessed it, and so forth. But you can actually look at we believe, microbiome risk assessment for obesity and weight gain, that there are differences in composition that you may be able to see, changes or detect patterns of change that would indicate one's risk for obesity. The same with cardiovascular disease and other diseases as well. There's been a lot of interest with colon cancer or Crohn's disease, the intestinal diseases, that I've been looking at literature for. And then obviously with medication, and in particular with surgery, bariatric surgery, for individuals with obesity, that there are changes in the gut microbiome that may have effects on other interventions that the individual is taking. There's much more recognition to ask and inquire about social health determinants. What's your home life like? What do you do for a living? What exposures do you have that could affect various levels of risk, from your developing obesity, your ability to intervene with a behavior modification, or is surgery your best option, and using that type of information to inform medical management. The same is true for disease risk, and trying to understand what that patient's environment is like and the likelihood of succeeding with one intervention versus another, as well as looking at their access to medications. Maybe they're 50 miles from a pharmacy. Maybe they are financially constrained, that's going to impact whether or not they are able to take their meds as prescribed. Maybe they have cognitive functions. Maybe they don't have family members around to help them take the 5 meds they need to take daily. And so risk of harm or adverse events may increase in certain environments. So more providers asking more about some of these social health determinants, and then more specific testing that could be done once a disease is diagnosed, so very specific measures. You're out of the primary care setting now into the specialty setting, doing more medication monitoring for certain therapeutics that may have a narrow window that require to make sure that you are optimizing outcome and reducing potential adverse events. So there is a gentleman that some of you may know from Stanford University, by Michael Snyder, who years ago came up with this, what he called it, more of a comprehensive assessment, using all these quote-unquote omics technologies to take stock of the different things that were going on in different systems. So starting with the DNA-based whole genome or whole exome sequence, looking at gene expression over a series of time points. I think the first paper was done over a year. So there was a lot of sample collection and a lot of data processing and analyses. And then also looking at the microbiome and the metabolome. So those are 2 different technologies that are used. And you're looking at 2 different things, even though they're interrelated. You're trying to understand differences in microbiome composition and diversity, especially as you have environmental changes. You're sick, you didn't eat one day, things that you had a sleepless night, stresses, and all these things that may impact the microbiome. And are they, can they be detected, and also in the metabolome and the potential effects of that. And so really just doing a comprehensive assessment of himself and some other individuals over a long period of time. So right now that's not feasible, really. I mean, I won't say not totally feasible. There are some people who could participate in those types of studies or live in that type of environment, the quantifiable self, where you're constantly taking measurements again and again, and you have an unbelievably large data set of yourself, and it can begin to detect patterns of changes that may be predictive of risk to your health or to how you may respond to a medication or whatever the case may be. So doing this type of assessment, there's a lot of repeats, which would take an enormous toll on the health system. That we do repeats. Every year you should be checking your cholesterol, or you're checking your lipids. Every day you may be checking your weight. So there's certain things we do repeatedly, and other things that are once. So your genetic risk assessment could potentially be once, so unlikely your genes may not change, the presence or absence of a variant may not change, this interpretation is going to change, especially in the times that we live in now. We don't quite understand everything with the detection of an A in one person and a T in another. The more functional assays, or the gene expression, the microbiome, or the metabolome, those are things that are so sensitive to the environment and what's going on that it would require regular monitoring. Some see differences in the daily monitoring. Environmental risk, that's something that is taken into account on every office visit, changes in your lifestyle, you become unemployed, lost a family member, or your support system, and lots of those factors would be documented in that case. And then your repeated measurements as required for any disease-specific or intervention-specific monitoring that's indicated. So the current health systems couldn't sustain that. That's way too many tests, not to mention the expense and the fact that I'm getting way ahead of myself and that we don't know what most of that means. We're in a stage now where we can measure lots of things. We can measure it somewhat accurately. The rapidity, that'll increase, as well as the accuracy. The price will come down, as we've all seen for those requirements for DNA-based sequencing. And that would put such a huge burden on the health system that you almost would have to share that burden with patients. And it's, there are examples where it's already occurring, so it's not like there isn't a precedent. There just isn't one with this type of technology or these types of data sets of gene expression or transcriptome or metabolome or microbiome, most of which is totally unfamiliar to the general public. So how could we incorporate a lot of this in? As I said, there are lots of patient devices and tools that patients do themselves, that they perform themselves, and that are able to are easy to understand. They're rapid, they're accurate, and they're cheap. So this is just a smattering of those you're probably aware of. Everything is connected to our phone via Bluetooth, so it's a lot of much easier data communication with a potential provider if you can designate one. I mean, some of these you do, some of these you don't. I don't think the pregnancy test or the ovulation test needs to be a provider, but you can actually track your results, and you can keep, of course, there's an app for everything, and see when you last tested yourself and what the results were and so forth. The watch, so the watches are only getting more complicated and more complex. These are the wearables. So Apple Watch is a big one, but there's other companies that have watches that are getting more and more complex. Some of them are getting FDA approval to measure certain types of measurements. Lots of different other systems, glucose monitoring, all this connected to Bluetooth. If you don't have a scale that's connected, your weight can be reported right into your EMR daily. That's available. Asthma inhaler. Again, you can track when you took it and so forth, and have that whole history documented and shared with your provider. So lots of tools that are already available. Now, can we imagine genetics and genomics making that jump and getting into one of these tools? So the technologists and the engineers are designing smaller and smaller sequencers. Can you see the same happening for a mass spec? That would be a bit of work, but it's certainly not out of the realm that we can use a lot of nanotechnology, or maybe change the spectrum or the scope of what's being analyzed to a certain number of fatty acids or carbs or whatever the case may be, that are devices that can be used at home, and that information can be stored and transmitted to the clinician's office. And so when a spike occurs or something abnormal or something out of the normal range occurs, that it could be flagged and followed up by the physician potentially if there's a need for an office visit. So a lot of us have been consumed, but how do you actually communicate information with patients about this? I see that. Yeah, 1 minute. So we actually have precedents. They may not be as successful. There's lots of data about how do we communicate scientific information, weather forecasts. There's actually quite a bit of literature of how you actually depict the trend of a hurricane or the path of a hurricane. And do people understand those cones? There's actually literature out there. Well, the same is true for medicine. How do we depict lots of complex data? And so technology has helped a lot. We can see lots of things, colors, graphics, and so forth. And there's a great deal of literature on this that we in the genetics and genomics community could certainly capitalize on and utilize. There's lots of ways to depict. This is more raw data. It doesn't tell you what it means, but it just tells you your answer of what you got on that glucometer or whatever the case may be. So I don't think it's out of left field that we can't figure out how to communicate genetics and genomics information. It's going to take some creativity, but there are examples that have been tested and work or resonate with certain individuals or certain education groups and so forth. So as I said, we don't know what it all means yet. So does that mean we need to reconfigure some of our ways or strategies or approaches to understanding the role of genetics and genomics in healthcare? So we have lots of groups, large groups, collaboratives that are looking at genetics and electronic medical records or genetics and sequencing and trying to understand different pathogenic classifications of certain variants. We haven't drawn all the partnerships between one group and another, and so we're still the silos are less, but they're still silos, and how do we combine some of these groups when we know we're going to need to look at multiple data sets that require multiple types of technologies and multiple analytical expertise and towards machine learning and AI. So bringing those into our field or our home or community of genetics and genomics, I think definitely needs to occur before any of this quantifiable self can actually take place. And so there's lots of challenges to that. And I won't go into that. I think most of us know this is tough. It's difficult, and there's lots of things to work out that may impede or slow bringing together even more large groups that currently exist to create even large groups, if that's the path forward to understanding how all these things factor into predicting drug response or disease risk and so forth. So back to the conclusion slide, which is the same as the second slide. So I thank you for your attention. I'm usually on autopilot for 75 minutes, so I can keep on going, but I know the time is up. So I appreciate your coming today. Thank you.
We'll just leave because it is 1 o'clock. Totally understandable. I think we'll just stay after and ask a few questions. Do you mind taking questions for 10 minutes? You can do that. I'm sorry we're over time. Though. Any questions? Want to manage your own questions? Please feel free to go if you have to. I apologize.
RNA data or the expression data. Or are there other ideas in that way to analyze like?
That's a question. I don't know what he did specifically. I'd say it was not a blood sample that he took from himself or from the participants in that small trial, daily or weekly. I'm not sure what it would be, but it's a good question of what would be the best sample that would be representative and most informative, especially with respect to gene expression, which is so tissue specific. So if it's the gut, I don't know. If stool samples is the easiest to do different types of analysis, both the microbiome or there's another tissue source that would be more informative. So it's part of working it out, and it's got to be easy if you expect patients to do this on a regular basis, especially at home. So there's a project at the School of Engineering, and you may have heard of it or others are doing it. It's called Smart Toilet. Lots of things that can go on. A lot of people have qualms about the collection of the samples, and so this is part of the drive around that, kind of to automate it in that it would be processed without your involvement, so to speak. So that's, with the colon cancer screening test and so forth, that's one of the reasons why people don't do it. And so the engineers are trying to figure out a way to collect it, to store it, and get it to a place that can be transported if it's to the laboratory with minimal human involvement, whether that's urine or it's stool.
So let me ask you a question. I think you may have questions about this. In terms of talking about pharmacogenomics and public health, I kind of expected that you'd be doing what you did partly, which was to say, in this overall picture of public health, where does pharmacogenomics fit in? And you know as well as anybody, better than most, the difficulty of even implementing simple testing, which seems so simple and so obviously positive. But then you went into these very complex things with no proven utility, which take, refiguring our toilets around the world.
Yes.
So isn't this almost like the wrong direction, given what you know about pharmacogenomics, or is this sort of the way things are going and it can't be resisted? Or what are the lessons we can learn from the challenges of implementing pharmacogenomics, or thinking about these other technologies too, which we're obviously drawn to as part of the overall picture?
Right, and you may be completely right. And part of this that came up with, you look at a paper, you look at the study demographics, weight's not always reported, and you get age, you get race, you get some diagnosis, but age does not always reach that level where it's a standard factor to reporting. And then you're looking at microbiome, and then the question is, well, how did something else affect that? You didn't even look at any pharmacogenetic variants or any metabolism phenotypes or anything like that. But you're looking at one thing at a time and trying to make an association, and we're not always seeing a strong association because there's so many other confounding factors. So I always tell the students, let's look at who you studied first. You can look at the conclusions, and they're all fine and good, but tell me what was the patient population like? And did they really do a deep dive into some of these other factors that we have an inkling, like weight, are going to affect medication response? Do I see a spectrum in weight? This stuff wasn't even reported in many papers. You just don't even see it. I see African-American, white, age, and diagnosis. Maybe medication history. Well, what about their weight? It changes so much with the PK. The metabolism, we just, it's almost overlooked. And then you just don't know how to interpret it. You don't know how to analyze it. So it's where all the analysts and the statisticians, and they get excited or maybe dreading, this is an ungodly huge size of data, and how to make those connections, a 1-to-1 and then 1-to-2. That everyone's an N of 1, essentially. That when you add enough factors together, we're all unique. But can we see trends that would signal a certain microbiome composition with a certain weight, with a certain pharmacogenomic profile is going to be X. So I don't know what the right approach is, but you could see the limitations that we still have, even though the technologies are growing around us. How do we keep bringing it together and not, I'm looking at this disease and microbiome.
I'm trying to think of how we study this to figure out. Different ways to talk more about approach.
Right, and I don't know if I was beginning to think that we're overthinking the issue. We all undergo lots of tests, Chem7 or any of these other standard tests, most of which I don't know anything about. I'm only told what's abnormal or falls outside the range, but I don't get a spectrum, I don't get a profile, I don't get a fancy snapshot of my, if you looked at, I often use LabCorp, and it's pretty static, it's all text-based. I don't even get color on mine. So, do we need, how much do we need is the question of how much do we need to communicate to patients to get them to a level, to an actionable level, or to be engaged in recognizing the importance of monitoring themselves on a regular basis. So as I said, star 1, star 4, that's great. We're excited about it. We know what it means. We know what the functions changes, but do the patients even need to know that? And I know I'm way over time, but yeah, I'm a human geneticist, so we go through all the clinical rotations, and I remember the genetic counseling rotations, and they would use a flip chart 20 years ago, and they would go through, this is prenatal, and you start with the chromosomes, and you can already see the glaze starting over, and you're like, do I need to tell them that we are doing a chromosomal test, but do I need to actually show them the chromosomes and they're 1 through 2022, X and Y, and you've just lost them. As soon as you open the flip book, and I'm watching because I don't have to communicate as an observer, but it's like, how much do we need to know? And it's kind of like the consent process. How much do you actually need to tell them about the details of the science to get them to understand what the purpose of the test is, or what the outcomes and how they could be used? And that I have high iron. That's probably all you need to know. And you need to watch your diet, or you need to take something. Can we use apply the same lessons learned to genetics? When we did focus groups, nobody likes the name metabolizer, and I definitely don't want to be called a poor metabolizer. That sounds horrible, and I don't want that on my record, and then it just sounds bad. And if anything, I'm not taking drugs. And then we had, you can't call a patient that. And we had one woman just came out and said that. I'm like, then how do we tell you? How do we tell you that your genetics will cause you to metabolize something a little bit slower? So we're going to adjust the dose. And she's like, that's it. That's all you need to say. Don't give it a name, especially when sounds really negative and scary. So it's kind of interesting to understand. People may not necessarily want the stuff we think they want. And physicians don't want the stuff that's on that report. And we've been told that, just give me the back page. What do? I don't care about the rest. I don't need to know the rest because it's not going to affect my judgment or my recommendations. Maybe it's the same with people. If you want more, you can always find it. There's no need to push it out. And I think it's the same thing with the phone and the wearables. There's lots of data you can look at. They're only going to put what you are today. If you want to go back and look at your history, or you want to look at certain types of data, you can go back and look at it or get more data. So I guess it's managing the control of information, or controlling the information, but allowing them to adjust the depth of information. I'm doing my fingers, and my children are just, they touch everything and they can change everything, and it's just at the touch of, if it's not on the phone, it's not going to work. So.
Thank you so much.
Great, thank you.